The one down there just was not well equipped for this kind of class. There's cardboard taped to the tops of the tables. There's like a roped off area that has like carcinogenic things in it. So I said, no, we're gonna go in here instead. This one's not awesome either, but it's better. It's like old school. You got chalkboard in the back. But we're gonna try it. If we find out this one doesn't work, then I will try and request a different one. But they are hard to come by for biology because we don't have most of our own classrooms in Brick Hall. So let's try it. We'll see if this one works out for us. Now we're in a plant taxonomy class, and I'm going to talk about two different topics that are somewhat related to each other in our first chapter, which is an introduction to taxonomy and systematics. They kind of go hand in hand because the new way of doing taxonomy these days is to use the information that we learned through a discipline called systematics to classify an organism and name organisms. And so there's a traditional way to do taxonomy, which is what we oftentimes use in the field. Easier techniques, looking at organisms and just looking for similar features like the same flower color to classify and group organisms. But what we're going to find is that's often not how they're related to each other. And nowadays we're trying to classify things based on their relatedness in terms of their evolutionary history, which is where systematics comes into play. We'll do more traditional taxonomy in lab, right? Because that's what we really need to be able to do to keep plants out, to identify them, and site recognition, those sorts of things. But we're going to be doing more systematics in lecture, so you can understand how the groups of plants are classified today and where this field of study is really going to. So let's talk about taxonomy first. What is it? This is obviously a plant taxonomy class. Well, taxonomy has basically four main activities. You are describing, identifying, naming, and classifying organisms. And we'll briefly go through what each of those things mean, even though some of it seems like it should be fairly obvious as to what that is. We're going to use classic techniques. We're going to use modern techniques. So one of the chapters that we're going to discuss in the class is going to be molecular systematics, okay, which means we're going to be dealing with DNA and using the DNA to figure out how we should sort and organize plants. Probably a topic many of us have never had before, or if we've had it, it's been a while since we've seen it. I don't oftentimes get a lot of the cell molecular students in here, and many of them haven't taken molecular, which is going to be very relevant to that course. I promise it's a short lecture, but you do still need to be familiar with it, because as I said, that's kind of where things are going nowadays, is using molecular data to classify and organize these different types of organisms. So we'll briefly discuss those. The goals of plant taxonomy is to, specifically plant taxonomy, is to recognize and explain plant variation, right? Differences between different species, even differences that can occur within a species, right? And to systematically organize that information into a way that researchers like ourselves can utilize it in a useful way. And so there's a couple different ways that we can actually take all this information that we're going to learn in taxonomy and organize it into a way that, into references and resources that people like us can use. And we'll discuss those in a little bit of detail as well. Now, understand taxonomy is going to be kind of a unique field of study. Right? Usually when we're talking about biology, we have all these subdisciplines like physiology and genetics, molecular biology, cell biology, focusing on very specific topics. But taxonomy is different than all of those other fields of study because it's going to take information from every single one of those fields of study all together and try and interpret it as a whole to determine the relationships between these organisms. So we're going to be delving into some pretty diverse forms of information. We are going to be talking about physiology. We're going to be delving into some of the biochemistry. That's how some of the construction of the cell wall, for example, is how some of the families are organized. We're going to be looking a lot at morphology and anatomy. We'll be discussing things related to evolution, genetics, right, and ecology. All of these things are going to come into play when it comes to classifying and organizing these organisms. And so you might be delving into a few topics that you may be a little bit less familiar with. Now, the basic activities of taxonomy, like I said, are fourfold. You have describing plants, you have identifying organisms, you have naming them, which is not done in just any old way. There's some specific set of rules that are in place for how to do this properly, and classifying. So we're going to run through each of these individually and talk about using plants as an example, how we kind of do this. Some of this chapter that I'm discussing may be a little bit of review from Biology 130. At least I'm hoping it is. Especially if you've had my Biology 130 class. You may see a couple slides that look very similar to what we talked about in that. Because it is very relevant to what we're discussing today. And taxonomy is something that we discuss in class as well. So let's start with our basic activity number 1, which is describing organisms. When you're describing an organism, what you're doing is you are assigning a feature or an attribute. Something that an organism has through its genetics, it's inherited this. Okay? Features that are called characters. You're looking at those. And what you are then doing is comparing character states, or those characters, between different individuals, different species. Okay, well, what do I mean by character versus character state? Okay. Well, a character is going to be some sort of a feature that you inherit. Okay. And I'll use a pretty simplified one as an example for plants, a flower color. If you are an angiosperm, which is a type of flowering plant, you are going to have a flower. And that flower will have some sort of a color. We're not specifying what it is, it's just that is a feature that the flower will have. Every flower is going to have a flower color. So that is a character. Okay? Now the character state are basically the different forms that that specific character can take. So think about all the flowers that you know. You probably can think of many, many different colors that they can come in. You've got white, you've got blue, you've got red, you've got yellow, you have pink, you have purple. Every single one of those colors a flower can be is a character state. Okay, so if you are looking at a plant that's an angiosperm, you're looking at its flower, it will have one of these character states, one of these colors. Okay? Well, that seems pretty easy, right? We can work with that. We oftentimes use color for a lot of different things for classifying things because we can, in most cases, readily see that. The problem is when it comes to botany, when it comes to plants, sometimes vernacular language, simplified layman's language like this, purple, pink, doesn't always work, okay? There's a lot of terminology involved in this. Sometimes our everyday vocabulary is not sufficient for the description, and so we're gonna have to use some new terminology that we're gonna learn. If you've heard anything about this class, besides the fact that I'm sure you've heard it's hard, which it is, you've probably heard there's a lot of terms, and that's no joke. It is kind of like learning new language, okay? And the terminology for plants is very oftentimes different than it is for animals. So for example, you've probably heard of radial symmetry, bilateral symmetry in animals, how many planes you can cut an organism and get a mirror image. We don't use that in plants. We use zygomorphic and actinomorphic. Why? Because we like to complicate things. No, because we just have our own terms. And oftentimes terms will overlap with things that you've learned in other classes. For example, operculum. If you've taken ichthyology, you've probably learned that's something that's in a fish's ear, but it's not in a plant. It's the cap on the sporangia in a moss, right? So it's gonna be a little difficult sometimes to keep some of that straight. And my best advice for you in terms of the terminology is to keep up with it. Where are you gonna learn the most terms? In lab, okay? We're gonna have 5 whole labs that are nothing but terminology, that's it. And your midterm in lab is nothing but terminology, that's it, okay? And you'll have 50 stations with different things that are set at the stations that are asking a question. What is the leaf symmetry? What is the leaf shape? What is the leaf base? What is the position of the ovary, the flower? Right? What is the structure right here? It's all terms, and it's not multiple choice, right? So you've got to keep up with it. The reason I'm doing that all the way through the lab portion before we really even get to keying is because the more you learn the terms, the easier the keying is going to be for you, because that's what they use. You don't have to look up every term every time you turn around when you're trying to key a plant. Not only that, when we start getting into the families, I'm going to be using the terms with the assumption that you know them already. I'm not going to stop and define them, so you have to know them. So here's just an example right here. Another character that we could give to a plant is leaf shape. And you can't say circle, triangle, things like that. We don't use those terms. We use orbicular or deltoid. Right? So here are just some examples right here. We've got elliptical, lanceolate, ovate, linear, there's oblanceolate, there's obovate, obelliptic. There's all sorts of different types of shapes that we're going to talk about. You won't have to learn all of them, but I'm going to select the most common ones to make sure that you're familiar with those. So we'll get to those in class, in lab later on. All right, so understand there's going to be some terms involved with this, and we'll just have to keep up with that. Now let's talk about the second activity that we have in taxonomy, which is important, identification. Right? Identifying what an organism is that you might find in nature. Okay? Recognizing, first and foremost, that this particular object or this particular organism is a member of a group that has a name, okay? And associating it with that name of the group. You could be associating it with a family, you could be associating it with a species, it just depends, and what group are you associating with it. But you have to be able to identify it to be able to say, I know that this is this species, or I know it's this in this family right here, okay? Now sometimes you might run across a plant, right, or an organism that nobody has ever looked at before, and it's not even described in scientific literature, right? You can still identify this in another way. You can identify that that organism is new to science, and therefore it needs to be described and it needs to be named by somebody, hopefully you. If you discover a new species, that's a really good way to publish. I highly recommend that you get a chance to do that. If you get a chance to see something new, make sure you publish on it, because you'll get credit for that. So, but you still have to identify that nobody's seen this before, and that this isn't something that exists in the current scientific literature. So how do we do this? How do you identify something? Well, you look at the organism. You describe its characteristics. Does it have leaves? Does it not have leaves? Right? Does it have compound leaves? Does it have simple leaves? Right? What is the arrangement of those leaves? Right? What do the flowers look like if they have flowers? Compare it to the different groups. Right? This family doesn't have any flowers, so I know it's not this family. Okay, this one does, but it has the wrong color flower, so it's not that family. That's kind of how you get to that process. And sometimes it is a bit of a process of elimination. Why is it so important? And to be able to identify these different species of these different organisms. Because that ultimately is going to lead you to a wealth of knowledge about that organism, many of which you might actually really need to know. Okay? So let's take our little plant right here. What is this? Why might it be important for us to know what this is?
It may be poisonous.
There we go. This is poison ivy. Right? Knowing how to identify that can be very useful, right? Because it leads us to additional information. Probably the first and foremost, don't touch it. Right? Don't get into it. We need to be able to identify it though to do that, which is why poison ivy is going to be one of the very first plants that I teach you how to identify. Because we are going to run across it in our field sites, right? Kind of hard to avoid it. I get poison ivy every spring semester. That's my gift to you guys. I'm highly sensitive to it since I've been exposed to something called poisonwood, which is a tree that's in the same family down in Florida. So it's going to be out there. We're going to learn how to identify that, but that's useful information to know because it tells us all of this information about that plant. Don't touch it. This is where it's found. And this might be useful because a lot of people mix this up with something called poison oak. We don't have poison oak around here. People think we do. We don't. All we have is poison ivy. And there's actually two different species of poison oak. There's the western species and the eastern species, but we don't have either one of them here. Alright, so we know the distribution of the poison ivy is this area right here. You go out west, you're gonna find poison oak, not poison ivy. Okay? And all of this other information about it. It can grow along the ground, so it can look like a vine. It can also grow upright and look like a shrub. Climb into trees and look like the tree that's growing on. All useful information for identifying it, making sure you don't touch it. Tells you a little bit about the size it gets, when it flowers, when it fruits, all of this information that might be useful for wildlife management, for example. If this is something that an organism uses as a source of food, is it available? What time of year is it available? That can be useful information. You want to maintain it? Do you not want to maintain it? I don't want to maintain it in my yard, I know that. Okay? But other people might. Now, how do you identify things? Multiple ways. The one that everybody always wants to be able to do because it's the easiest one is something called site recognition. Meaning you see it on site, you're walking up to it, you look at it, you know it. You automatically already know what it is. Okay? Now, I'm going to teach you to site recognize some things in lab, but I cannot teach you every single plant here in Tennessee. Okay? There are way too many of them. It's not like herpetology. It's not like mammalogy where you've got a limited number of organisms and you can get through all of them in a semester. There's no possible way I can teach you all the plants in Tennessee. There are just too many. Okay? So what I'm going to do, and I don't even know all of them by site recognition either. Some of them I have to key out myself. So what we're going to do instead is we're going to pick and choose some of the more common ones, the ones you're most likely to run across, the ones that are going to be out and flowering during the time period that we're going to be having in class. Okay, so it can be relevant to your lab work. Just to also incorporate some of those example plants that we're going to see in the environment around us to utilize our second method of identification, which is to recognize major plant groups. Since I can't teach you to sight recognize every single species, what I can do is I can teach you to recognize a family, right? I don't know what species this is, but I know it's in the Apocynaceae family. I know it's a milkweed. I don't know what kind of milkweed it is, but I know it's a milkweed, right? That's useful because at least it narrows it down. And it gives you some place in the book to go and start looking for it. Gives you a place in the herbarium that you can go and look for it. It helps a little bit with the identification, so it's not quite so overwhelming. If you've never seen this plant at all, and you don't have the skills to key it out, you have no way of finding it out unless you go find somebody that does know it. So this is something that can be very useful. So we will spend a good bit of the semester specifically talking about families, right? So we can start to recognize some of those. There's only a few hundred families. No, we're not going to 100 families. We're going to be doing the most common ones. But there's only a few hundred of those where there's hundreds of thousands of species of plants. Okay? And just the ones that I teach you here in Tennessee doesn't mean if you go down to Florida or out west, you're going to see any of those. You get completely different suites of plants in different regions of the United States. When I went down to Florida as a postdoc, I knew maybe about 25% of the plants. All the rest of them I had to learn fresh and new because it's a subtropical environment, not temperate. Completely different plants. Out west, wouldn't have any idea until I started cleaning them out. They're completely different. Right? So, that brings us to our third method for identifying, using taxonomic keys. My guess is that some of you have already used a key before in other classes. Raise your hand if you have. Perfect. So we've got a little bit of familiarity with it. We also know how stressful and how annoying they can be sometimes, right? And I'll tell you right now, with plants, it's going to be 10 times worse because there's more plants than there are the other organisms you've been keying out so far. If you've seen your book for lab yet, that is just one state of plants, right? Imagine if you had all of North America, or if you had all of the Western Hemisphere, right? It's a lot of plants. And the first time you key this out, you're going to want to throw the book out the window, I promise you. But the more you do it, the easier it's going to get. Having a little bit of experience with other organisms is going to help. What is going to make this one unique and a little bit more difficult is that we don't know the terminology yet. So that's why learning the terms is so important. It's going to make your life easier when it gets to keying. Okay? This is the main way that we're going to be identifying plants. And I know a lot of us are going to try and use apps on our phones for this. I'm going to tell you right now, it's not allowed in lab. You cannot use any plant identification apps on your phone or any resource like that to identify plants for class. And you're going to have to do it the old-fashioned way. Those apps can be incredibly inaccurate. And if you don't have any other way of identifying it other than that, you don't know how to key, for example, your boat is sunk at that point. You have no other options. This right here, keying, is probably the most useful and most important thing that I will teach you in this class. It is the most important skill for you to take away from this class. So I want you to take it seriously. Okay? You're going to be doing some of this on your own outside class. And the more you do, the easier it's going to be and the more you're going to get out of it. You're only going to get out of it what you put into it. Okay? So I highly recommend that you do as much keying for practice as you possibly can. All right. So what do you do when you key? You emphasize differences and similarities between plants. Okay? It is a step-by-step process of elimination. You have to go step by step. Start with the first one. Okay, look at your options, figure out which one matches your plant. That's going to tell you which step to go to next. And go to that one. Okay? Look at the options, figure out which one matches your plants, and then go to that next step. Okay? It takes practice, particularly if you haven't done it with plants before. I'm going to teach you how to dissect plants in lab, because that will be something we have to do. And I know students tend to be a little bit hesitant to use the microscopes. They want to sit there and look at it like this. Please use microscopes. You know, you're going to go blind and go cross-eyed if you keep trying to look at the little plants that you're going to be looking at in the lab with just your finger, right? Make sure you look at the microscope. It's not the same as dissecting an animal. One thing, they're not going to run away and scream at you. Right? It is a delicate process, though. We're going to be using razor blades. Please don't cut yourselves. Don't want to do paperwork. And we're going to be using razor blades, we're going to be using little pins and things like that, but I'll teach you how to do it. And the more we practice with it, the easier it gets. Right? Now, there are different types of keys. Most keys are what we refer to as dichotomous. Now, that means that at each step, you are presented with just two possible choices. Two contrasting choices. Yes, there are keys out there that have more than 2 choices. You can have 3 choices, you can have 4 choices, but most keys that you're gonna see are gonna do just 2, and we call them dichotomous for that reason. They can be formatted differently though, and we'll talk about that in a second. But let's delve into what these steps are first before we get into that. Each pair of choices in a dichotomous key is referred to as a couplet. Right? So for example, this would be a couplet right here, got 2 options. This would be a couplet right here, right? This would be a couplet, and so on and so forth. Each part of the couplet is going to compare equivalent but contrasting characters. Now, the first time everybody reads that sentence, they're all like, what does that mean? Right? Well, it means that, as you notice, in each one of these couplets right here, they all start with the same subject. This one right here is discussing the habit. Now, habitat, habit. That is an actual word. It means something for plants. Right? Habit, herb, habit, a shrub. Right? This one right here, they're both talking about margin of the leaf. Margin of the leaf. That is their subject. You don't have one part of the couplet here saying the margin of the leaf is this and the flower is this. Why are we talking about flowers? We were just talking about the leaf. You need to always be looking at the leaf. That subject should be the same for both of the choices in that couplet. So that's what it means by it's comparing equivalent, margin of leaf, but contrasting characters. The margin of this leaf is either serrate or dentate. The margin of these leaves are entire. Okay? So that's why they're contrasting. One part of the couplet is going to be accepted, the other is going to be rejected, always. Which means one couplet should match your plant that you're looking at and one of them should not. If both of them don't match your plant, what's probably wrong? You're not in the right place in the key, right? Okay? If you get to a couplet and neither one of these 2 choices make sense, you've gone off somewhere and you're gonna have to go back. You're not in the right place. Because it will always have one couplet that is correct, that matches your plant, and one that is rejected. That's the only way you can figure out what step to get to next. You follow the pathway that goes with the couplet part that is accepted. Right? Now, we have different formats that dichotomous keys can take. It's a matter of preference as to how you want to organize these couplets, what you want them to look like in your book. So here's one example right here. This is something called an indented key. And the reason it's called an indented is because you can see not all the lines start in the same place on the left side of the page. Right? You'll see that each couplet is indented a certain distance inward as you get further and further into the couplets, into the steps right here. So our first option right here, habit is a tree, habit is an herb, that's the one that's furthest to the left. Right? Our next option beyond that then is the next one that's going to be indented. This is in right here. So we've got plant is evergreen, plant is deciduous. If we took this path, it would be this one right here. Leaf arrangement opposite, leaf arrangement alternate. Okay? Now you notice for mine right here, I put numbers in here. And oftentimes that helps a little bit with organization, but they do not have to do that. I have absolutely looked at indented keys before that have no numbers in them at all. Okay? That's not a requirement. Okay? I think the one in your lab does have those. Okay? But just be aware that there may be some that you run across in the future in your future careers that may not have these numbers. It doesn't have to have that. Why is that important? Because the next type of what I'm going to talk about next, I'm going to talk about, does have to have the numbers. They're absolutely essential. Right? So this one, the importance is that they indent them different distances. Now there's some advantages and disadvantages to both of these types of formats we're going to talk about. One of the nice things about indented keys is that it groups together similar plants that have similar characteristics. Right? So as an example here, I'm having trouble keying this plant. I can't figure out what it is, but I do know that it's an herb. Okay? Well, that means that I can eliminate everything up here because the herbs don't start until down here, so only these right here are herbs. These are all trees, so I can just ignore those. And maybe I can narrow it down a little bit more with these down here. Right? That's super handy. Right? But there's some disadvantages to this type of key too. One, it is a waste of paper because you're losing a lot of white space here, and you can end up having a much longer key if you do it this way than if you do the other method we're going to look at. Right? We're trying to be economical and ecologically nice about this. We would probably not do that. Right? The other thing about this, besides the fact that we have this nice arrangement of these plants being similar to each other, being right next to each other, The couplet parts you can see are not right next to each other, right? This is the same couplet, plant a tree, plant an herb. Those are your two choices, but they don't have to be on the same page, right? Because if you've got a lot of information between here that has to do with trees, this could go on and on and on and on, and you may not get the second half of the couplet until 2 pages later. So you have to sit there and go through until you can figure out where the right indented one is to go with the other half of this. That can be a little bit confusing sometimes. So you could go with this option instead. You can use a bracketed key. Now for a bracketed key, the two parts of the couplet are always going to be right next to each other in consecutive lines. Super nice. You always know where the two parts are going to be. They're always right next to each other. Okay? So this is our couplet right here. That's one. This is our next one right here. There's one right here. We know that those go together. So it helps a little bit with organization. You can see that there's no indenting. So we're not wasting paper or anything like that. But you have a disadvantage related to this. That was kind of the advantage of the previous one. Before, I could kind of use the process of elimination and say, okay, I know my plant's not a tree, it's an herb, so I know it's got to be down here somewhere. And those are the ones that are herbs, so I can eliminate these other ones. And these bracketed keys don't organize this the same way. Okay? This is how it's organized. Each one of the lines will end in either a name. Now that name could be a family, could be a genus, or in this case a species. Okay? Or you'll see the other option is to end in a number. Now what that number is it's referring to a couplet later in the key, and that's your path of where you're supposed to go. So for example here, if you have a leaf that has, or plant that has leaf arrangement alternate, you would go then do couplet number 6. So we jump down here, that's our next option. However, if you were having a plant here that has a leaf arrangement that's opposite, you would skip 6 and you would jump all the way down to number 7, and that would be your next option. And that would be your next step. So you have to kind of keep track of your numbers and figure out where you're going. But what that means is that we're not grouping similar plants together. Okay? So if you're trying to, through process of elimination, you say, okay, I'm looking for, I know my plant has serrate or dentate leaf margins. Okay? If you were in a indented key, you could say, okay, that means that everything underneath here is serrate and dentate, I can look at those. But that's not actually the case. Because you'll see leaves that have entire margins are stuck in the middle right here. And we have other things that are also entire margins right here. So all of these are entire margins. You don't actually get down into your serrate and dentate ones until you get further down. Right? So it's harder to do process of elimination. Advantages and disadvantages to both. I do want you to be able to recognize the two different types. So if you're presented a key, you should be able to recognize it. Is it dichotomous or not? Is it indented or is it bracketed? Later on, we're going to do a homework assignment in lab that you're actually going to make a bracketed key. So that's kind of an important one to be able to recognize for that reason. In your lab, you're going to be using indented keys. So that's why it's important to know those. That's why I'm introducing both of those to you. All right. Our next step in taxonomy is to name things. We're naming organisms. That means we're going to assign a code, a name, that stands for an object or for a concept. In this case, an organism. And what is the name that goes with that organism? And again, this could be a species name, could be a genus name, family name, kingdom name, domain, all of those apply here. And we'll get into that in just a little bit when we get into classification. Now, that doesn't mean you can give an organism just any little name. Okay? It is absolutely essential for communication that everybody understands and is familiar with all of the same names, which means we have to make sure we're all using the same language, okay, that everybody's going to understand, okay, and that we're following a set of rules that everybody follows. Okay. So for example, we're going to find out that scientific names have to have two parts to them. Okay? You can't name something with only one name. That's not a valid scientific name and nobody's going to recognize it. It has to have two parts to it. That's one of the rules. Right? It's going to be in Latin. That was the language of the sciences in the past. Nobody speaks it now, so if anybody tells you that you're pronouncing it wrong, tell them you don't know that because nobody speaks it. Right? So we're going to kind of speak it how we want to help us remember how to spell it, because spelling is going to count in lab. Okay, so just come up with little phrases and things to help you remember those. But we're going to be using Latin names because that's part of nomenclature. Nomenclature is a way of naming organism that follows a set of rules. we're gonna have a whole PowerPoint on this. It is not the most exciting PowerPoint. I acknowledge that, right? But it is important to understand how plants are named and that they're not named the same way that bacteria are, the way that animals are, or even cultivated plants for that reason. We're going to talk about that as well. And one of the reasons for that is it's going to have to do with one of your big projects in lab, which is going to be your collections. So that when you're doing the collections and you're writing your labels and things like that, you're going to understand the format you're supposed to be using. Because if you don't use the right format, then you lose points. So we'll talk about what all these parts mean, what this is going to be related to. And so we'll get into that in the next lecture. What we're going to talk about next, though, is classification. Activity number 4 in taxonomy. There are 2 basic processes involved in classifying organisms. This hopefully should be reviewed, right, from what you learned back in Biology 130 or even 140 for that matter. Right? You're going to group organisms by sorting them into groups that have some sort of common feature. Right? So, for example, we group together our black bear here and our wolf down here because they're both carnivores. They eat meat. Right? They're also mammals, which means they're going to have things like mammary glands, fur, hair, those sorts of things. They're in the group Chordata, which means that they have 4 features during some point in their life: dorsal hollow nerve cord, notochord, pharyngeal slits, posterior tail, all those. They're also an animal. Right? So they have the features of an animal, and they are eukaryotes, and eukaryotic cells. So we group those together because they have similar features. But you'll also notice that they don't fall in all the same categories. As we get further down here, the wolf disappears because it is not a bear. Right? And we'll get into what these categories are. Right? But you'll notice that the category, the number of species in each of these categories keeps getting smaller and smaller and smaller and smaller. That's referred to as ranking. Organizing the smaller groups within progressively larger and larger groups. These are the ones that are more inclusive because they have more species. These down here are more exclusive because they have fewer species. And think of it like a country club. If it's exclusive, only a few people can become members of it. If it's inclusive, a lot of people can be members of it. Species is the most exclusive because there's only one species. Now these ranks should be familiar. We learned those, I know, in Biology 130 lab, or at least the ones that are listed there. And so we're going to go back and kind of refresh your memory on that. Each one of these ranks, these categories that we're talking about that we use to organize and classify organisms, are referred to as a taxon. This is a taxon, this is a taxon, that's a taxon. When we're talking about multiple ones, we'd say taxa. Okay? So that's a group of organisms of any rank in the classification hierarchy. But we have specific names or terms that we use for each one of the different taxa. So we've got domain, kingdom, phylum or division, class, order, family, genus, species. You'll notice I have some of these in bold and some of them are not bold. The reason for that is because some ranks, the ones that are in bold, are always assigned to a plant. Every plant out there has a species, a genus, a family, order, class, division, kingdom, and domain. Not every plant, though, has a subspecies or a variety or a subgenus or so on and so forth. So the only ones I want you to memorize are the ones that are bolded. Know the ones that are bolded and know the order. You probably in 130 came up with a little phrase or something to help you with that. So something like, dear King Philip came over for grape soda. Come up with whatever you want to. The students come up with way better ones than I do that help them remember it way better than mine would. But whatever works for you to remember the order of those. You're probably wondering about this right here, division phylum, okay? Because we typically learn that as phylum, which is what we use for animals. But botanists, again, we like to complicate things. We don't generally say phylum, we say division instead. It means the same thing, right? Division is a phylum for plants. Okay, now, if the species or an individual plant does have one of these other ones, it's only been assigned because it's needed. Sometimes you need to have a subspecies or a variety. You'll see that occasionally in your book, as we're keying some of the plants out for your collection, you may run across a couple that has a variety or subspecies. So here's just an example of one you might run across. There are lots of different types of violets. Violets are a pain in the neck to key out. But I'll give you some extra resources to help you with those if you decide to do those, because it is one of the ones that's gonna be out when we're doing our collections. But violets are notorious for looking very similar to each other, right? And this one right here, these two plants right here, they are in fact the same species. Both of them are Viola soriae, and they both are the same species. But this one here is considered a different variety than this one is right here. This one's variety soriae, this one's variety missouriensis. The only real big difference between them is that this one has a little bit more rounded, more liver-shaped leaves. These have a little bit more triangular leaves. These are gonna have furry surfaces on their leaves. They're kind of fuzzy feeling. And these over here are not having hairs on the leaves. That's what glabrous mean. And these are typically gonna be found kind of all over a variety of habitats. They're kind of disturbance plants, weeds, if you would. And these right here are frequently found mainly in wet woods. Okay, so that's what distinguishes those varieties. But it's not big enough differences for them to be classified, according to the scientists that have described these, to be considered different species. All right, now, there's a lot of information that we're collecting about these organisms. And we're determining their identity, we're looking at their characteristics, determining when they flower, when they fruit, what their leaves look like, what the margins of the leaves are, the shapes, color of the flower, all this distribution, where they're found. That's a lot of information. And for every single plant that's out there, that's not something that any one person could possibly hold in their memory or pass on to another person for that matter. So we gotta keep records here. And it has to be kept in a way that other people can use it and it can last the test of time. So we have over 300,000 plant species that are cataloged, but we're constantly finding new ones. Not only that, we're also finding new information about the ones that we already know about. So adding to the record doesn't necessarily mean you have to find a new species or find a new plant. You can still add to the record the information out there about already existing species that we know about. You just find more information about them that we didn't know. We thought this plant only flowered from May until June, but we're starting to see herbarium specimens now where we're starting to see flowers in them in April. So maybe that range needs to be expanded. We can revise this, we can add to this as new information is gathered. Periodic updating is really necessary. And now we're starting to get kind of a wealth of revisions, right? Because we have this new digital world. Now all these specimens that used to be locked away at one university that very few people had access to are now all digitized, photographed, and entered into portals and different online websites that you can access. And now researchers from across the globe can look at these and provide additional information that can help with this. And so we're starting to see a lot of that now with all this digitization. Our herbarium just got digitized a few years back, right? So we're now part of that world. It's actually pretty exciting. Now, records. Records oftentimes are going to be in the form of something called floras or manuals. So your book, for example, that you have for lab, the Flora of Kentucky, that would be one of those. Inside of it, you'll find a lot of different things. One of the things you're going to find are keys, okay, for basically keying out the local flora of that region. Usually these floras are not going to be over a large area. Usually it's a state or maybe the southeast. Like every once in a while you'll find one for like North America and the United States, but most of them are usually smaller than that and have a specific region or a time period that they focus on because there's so many plants out there. You start getting more and more of those, it's harder and harder to separate them out even with the key. Right? But the aim of these is to allow identification. Okay? Now what's the main difference between manuals and floras? Mainly the amount of information that's included. Both of them are going to have keys that allow you to identify plants for that region. But manuals tend to have some additional stuff in them. They usually have descriptions of the genera, they have descriptions for the families, even for the individual species. They tend to be a lot thicker and a lot bigger for that reason. A lot of useful information. So where does all this information come from? How do we know that this plant only flowers from April to June? Because obviously this person hasn't gone out into nature, just wandered around and found every population out there of this plant to find out and watched it to figure out when it flowers. Okay? Well, they get this information from individual specimens that people have actually collected at different time periods, different times of the year, different years, in different places. So this is gonna bring us to another one of our specialized records that you have for plants. Has anybody taken entomology? Yes, perfect, okay, so you're familiar with insect collections, right? So we have an insect collection on campus as well. It's the same idea here. You're going out, you're collecting specimens during a certain time of year, and make sure you've got information on that specimen that gives you who collected it, when they collected it, where they collected it, what time, all that sort of stuff. You do the same thing for plants. Okay? But how do you do this in a way that you can preserve this for the long term? Because this is something that needs to be preserved forever, right? Not just for a couple of years. This is something that should last forever if it's kept properly. Okay? So what we refer to them as are herbarium. Herbariums are going to be a collection of preserved plants that are stored, cataloged, and arranged systematically for study. Usually they're organized by family, and oftentimes those families will either be in alphabetical order, or they will be in order of their evolutionary history, which is how ours is organized. Inside of them, you'll find thousands to hundreds of thousands of preserved specimens. So when you collect, and you're gonna do a collection in a lab, that you'll get a feel for how these specimens are actually made and the proper way to do them. When you do this, you have to preserve them in a specific way. So for example, you have to have at least one leaf turned upside down, so that when it's glued on the page, you can see the surface features on both the top and the bottom of the leaf. Sometimes they're different. You have to have enough of the plant that you can see the arrangement of the leaves. It needs to be reproductive. You need to have all of this information down here about when you collected it, where you collected it, who you collected it with, what other plants were around there, what the habitat was like when you collected this. All of that is the information that people are then going to look at for all the specimens for this species to figure out what to write in a manual or flora for that species. And every plant that they've ever seen that had flowers on it was collected sometime from April to June. They know that's its flowering period. So this is important for it to be accurate. Don't lie on your labels. Okay? Be accurate about the information because it will be used. Some of yours will actually be put into the herbarium at the University of Tennessee Lark. Okay? So you want to be accurate about that. And we'll talk more about that in class and then we'll get there. I'm going to pass these around though, so you can get a chance to see what some of these look like and how detailed the information should be on those. And there are special types of specimens as well. And there has to be a specimen or a plant out there that somebody collected for the very first time that nobody's ever seen before, called type specimens. We're going to get into that in the lecture after this one. I will tell you that we are not fancy enough school to have any of those. But if you go to UT Knoxville or some of the bigger R1 institutions, you're probably going to find some of those there. There are specimens though that are herbarium that date before I was born. So I guarantee before you guys were born as well. So if you keep them properly, they'll last forever, and you can continue to get more and more information from them. So this is an important type of record for plants to have, these herbaria, across the regions. All right, now that we've talked a little bit about traditional taxonomy, now we're going to get into a topic that relates a little bit more to how we classify and organize plants today, which is going to be more the focus of the lecture for this semester. We're getting into systematics. This is another subdiscipline of biology that specifically studies the diversity of life, both past and present, so species that existed in the past that are extinct now, ones that are existing today, and the relationships among living things through time, right? How they're related through their evolutionary history. Why this is important is because this kind of information, we're going to be taking it nowadays and using that information to classify organisms as opposed to just obscure characteristics that we just happen to look at that we think are easy to see. Because a lot of organisms can look similar and not be closely related at all. And that's not how we're trying to classify them today. So this is going to delve a little bit into things like phylogenetic trees, which hopefully you had a little bit of that also in biology 130. If we've had evolution, or if we've had genetics, things like that, you may have seen some of these phylogenetic trees as well. You're going to see lots and lots of phylogenetic trees in my lecture, and you're going to want to know them. And I'll tell you the characteristics you'll need to know for those, and you have to understand how to read them. So if you don't remember from your previous classes when we talked about this, let's refresh a little bit on how to look at these. All right. The tools for studying evolutionary history include phylogenies. This is ultimately what we would like to know. We would like to know the evolutionary history of a specific group of organisms that we are interested in studying. The problem is, we can never know that. Never know it for sure. Because nobody has ever been there for the entire lives of these species in their existence. So we can't possibly know this for sure. All we can do is hypothesize based on the data that we have. And that data could be molecular based, DNA, it could be morphological based, anatomy, for example, things like that. That's what we have to draw from in terms of data to come up with this hypothesis. Usually we present our hypothesis as a branching diagram like you see right here that's called a phylogenetic tree. But understand, any phylogenetic tree that you see is a hypothesis, which means it can never be proved, and it can always be revised with more data. So if we've made a phylogenetic tree based on the data we currently have, but a couple years later we get some more data, new sequences of DNA that we can include in there, we realize, oh, that doesn't match up with what they had before, you can change it, right? But now your new tree needs to match both the current data that you just received, but also the old data as well. Just like any hypothesis, you have to revise it so that it matches all of the existing data then at that point. So we could never prove this. We never know if this is exactly how they're related. And then what's really fun is when you make a phylogenetic tree out of molecular data and one out of morphological and they don't match. And that happens frequently. So we'll get into that a little bit when we get into molecular systematics. So we're going to talk a bit about how to read a phylogenetic tree. Okay? Understand that phylogenetic trees are drawn along a time scale. Okay? So if you're up here at the top of the tree, keeping in mind that sometimes phylogenetic trees are turned sideways, but this one we've got it upright. Right up here on the top, this represents present day, the things that exist today, current time period, right now. As you move down in the tree, further and further down in the tree, you're going further and further into the past. So this point down here at the bottom of the tree is the furthest point in the past that this tree goes to, right? Doesn't mean you can't go further than that, but for this tree, that's as far back in the past that we go. So that's the oldest time period for the tree. That's something to keep in mind. The tree is then made up of a root, which is the very bottom down here. It's made up of lines, all the lines that you see right here. Nodes, which I've actually represented in this tree with the little green dots, but those are not usually there. They're just places where it branches, but these would be the nodes. Okay. There's multiple nodes. And clades. Clades are going to be groups of organisms, and we're going to talk about that in extensive detail. Okay, we're going to stop here for today. We'll get further into this as we get into our next class.